You're online. You do need a pointer? Or?
I have one.
Yes.
All right, thanks for the introduction. Yeah, you've pretty much said everything. I've worked on the evaluation of transmembrane helix prediction in the past, and since you've heard about these methods on Tuesday, I thought I'd give you some additional details about this. The date is wrong, but whatever. And I have a hard time judging if this will be too easy to you or too difficult, or if I'm going too fast, please shout at me and I'll slow down, because typically if no one does anything, I tend to talk very fast. Okay, so let's start. First of all, the question is, which methods do we even evaluate? So you might remember from the last lecture that there's a ton of high differability scales. They all differ, they kind of agree, they kind of don't, and all of these, in a way, you could consider their own prediction method. Then on top of that, of course, you have many methods from several decades of development that use advanced machine learning models such as neural networks, SVMs, HMMs, and so on. When I compiled the list, I found around 30, but even then it wasn't everything, and by now many years have passed, so we are probably around 50 or even more. And then on top of all of those, there's consensus predictors, which use single prediction methods and combine them and try to merge the output to, to get a better prediction, ideally. So which ones of those do you choose? First of all, why not take all of them? You might think very naively, if we can't decide, take all of them. That, unfortunately, is not going to work very well, because typically these methods are not as easy to get running as you would like. So it can range from anything between you just run it and it works, to you will spend the whole day changing the source code to your environment until it kind of works and then it still breaks at some point. That is, if you even get the method. Some of them are not available anymore, some never were, they were just published, but yes?
Why would you try to turn the source code to run in your environment instead of, say, just getting a virtual machine with something like Alt and
yes, that would help for some problems, but for example, if it was compiled 20 years ago on Fortran and the compiler simply doesn't exist anymore, the library changed, then this is not going to help you either. And then, yeah, of course, I mean, it's also kind of an overhead, right? I'm not sure if you would want 20 different virtual machines running, just one for every method. If it's just something of one use set shell, one use bash, then it's probably easier to change all the file redirects than setting up a virtual machine just for this. But yeah, you're right. Generally, especially if you don't have that many methods, that's also a valid approach. Okay, so that's one thing, and then of course the more methods you have, the more constraints you have on your input data. So not every method can predict every sequence. Length is a typical feature where they differ a lot. Some of them don't want to predict too short sequences, but some of them also break for very long sequences. So the more methods you have, the more constraints you have on the data that you can evaluate, and it also gets important for which parts of your data set you can consider new, but on this we will come back later on. So in summary, we can't really take all. We have to focus on some methods that are of particular interest. So which methods are of interest? Now, generally you could, so I mean in the papers everyone says we are the best, right? Otherwise they wouldn't get published probably, and even if not, they of course want to believe that they are the best. And they probably have good reasons to say so, but this doesn't really help you choose a method. So you could take recently published methods because they had access to a lot of new data, to the most amount of data. They also know about the most recent advancements in machine learning development and so on. But you could also additionally look at methods that are somewhat unusual. So we will see one method later on that was published very late in the middle of 2000s, I believe. And it went back to an approach that is more or less what people used to do at the very beginning. And initially this might seem like a bad idea. Why would you do this, right? But it turned out that by stripping down the whole problem to the bare minimum, they actually performed pretty well, and also got several advantages from this. So looking at kind of more creative, unusual approaches might actually be worth your while. But most importantly, you want to evaluate the methods that are actually being used. So just because it's published doesn't mean that anyone has looked at it before. And if, for example, you want to evaluate what, say, there are experimentalists that are using prediction methods to design their experiments in the red lab, right? And they probably chose their method 15 years ago when their protocol was established, and back then it was the best method, but that doesn't mean that's still the case these days. Maybe it is or maybe it isn't, but they are interested in this one method they are using is still good today. So this is probably the most important point, to look at what the most popular methods are and what the methods are that are being used, even if they are very old or, yeah. Okay, so next, one more thing, we have to compare against something. So what is our baseline? What if we make a predictor as stupid as possible, deliberately? What can we expect in terms of performance? So the most random predictor would be that something on the residue basis says, okay, this is a helix, this is not a helix, and so on. But that, of course, is not very good, right? Because you get helices that are 2 residues long, for example, and clearly that doesn't make any sense. So let's make it a bit more intelligent. Let's tell it, okay, helices are, for example, always 20 residues long, and randomly place segments of 20 residue long helices somewhere in the sequence. You can improve it even more and tell it, okay, I have a data set on which I'm going to evaluate, and I'm going to give you the information on how long the segments are in this data set. I give you distribution, and you randomly sample from that distribution, and then you have not a fixed length, but lengths varying between, say, 10 and 30 residues. So that gets you a bit closer. Next, you can also adjust the number of segments. So again, you might have many proteins that have only a single helix, for example, and much less that have 10. So again, you can build a distribution from the data set and tell this the predictor. Then according to this, let it predict the number of segments and the length as well. And this later on will appear as just random. So in the results we see this is one of the methods. And then one that's a bit more advanced is the semi-random, which gets told for every sequence, okay, you have to place, say, 3 helices here, only where it places them is random. So this already it's a pretty good advantage in a way for the predictor, but at the same time it's still a random prediction, because he can put it everywhere. Unless the sequence is so small that really only 3 fit, then it's a pretty big advantage. Okay, and finally, as we just heard again, hydrophobicity is the defining feature for transmembrane helix prediction. So let's just make a predictor that takes a window of 20 residues, and it sums up the hydrophobicity values over this window. And if that exceeds a certain arbitrary threshold, let's just say, okay, this is a helix. This is very close to what the earliest prediction methods did for transmembrane helices, and yeah, let's see how this performs, right? Because that everyone can build in half an hour, and yeah, this is very basic. Okay, so we have methods. Next, we need a data set. As you know by now, data for transmembrane proteins is very scarce for many reasons. Determining their structures is very hard, and generally they're not as nice as a soluble protein because they're always in the membrane. You kind of have trouble accessing them, and they're very nasty. In the past, there were these so-called low-resolution data. So through biochemical experiments, you can, for example, say label the loop on the outside or inside of the membrane, and then through that you know, okay, if this is outside, then the part in between kind of has to be transmembrane helix and something like that. However, it turned out that this low resolution data is typically quite error prone and actually is not much better than predicting from a prediction method. So clearly you don't want to evaluate a prediction method on what is essentially just the quality of a prediction method. So this is not really being done anymore.
Let me just interrupt you. So the way these experiments are done, imagine a plot has 3 membrane helices, and you put a fluorescent marker on this loop. Biologists can do that relatively easily, specifically. They can target a specific residue onto which they put a fluorescent marker. Now, you shine the light onto that. This is the outside, assume this is the outside membrane. You see the fluorescent light, you know it's this loop. You don't see it, you know it's this loop. Or on either of these 2 loops they can put the marker. That's the trick here. And that is what he calls low resolution experiment, right?
Yeah.
It gives you good light, it gives you good data if you don't know anything better, but it actually makes many mistakes for this.
Right, so these days we don't have to rely on this anymore. We have structures, right? It's very hard to determine them from transmembrane proteins, but it's possible, and there are some, especially by now we have a few. So let's take structures. Now this is a structure of transmembrane protein. Does that solve our problem? Do we know now where are the transmembrane helices? Where are they? Do we see it directly from the structure? What's the problem? What else would we want to see if we were to identify the transmembrane helices?
The membrane?
Exactly, and the membrane is never part of the crystal structure, right? So this is typically from X-ray crystallography. You build a crystal of protein, you have to extract the protein from the membrane. The membrane isn't there anymore, really. There might be some detergent, some small lipids that make this whole thing not collapse in itself, because of course it expects that here on the outside it's kind of hydrophobic because that's its native environment. But even so, and these small lipids and detergents, they are very flexible. You almost never see them in the crystal. Even if you have a near perfect crystal and even those are resolved, this is still not the native environment. So even if we had them, it's not too much help for us. So we don't really know. I mean, in this case, okay, there's not a whole lot of choice. This is like a poster transmembrane protein. I guess it has to be somewhere around here, right? But still, there are much more complicated ones, and it's not that easy, even from the structure, to say, okay, here and here is the helix. So, there, oh wait, I just said all of this, I believe. Yes, so let's skip it. This is just if you look at it again at home. So, there are several approaches to, from a given 3D structure, annotate where the helices are. And there are two big databases that are used here, OPM and PDBTM. And they differ because it just is not the one solution. And in the end,
Explain again what exactly they do.
yes. So again, they get something like this in the end. They get a structure, and then they look at it, and they have algorithms which I also cannot explain in detail and are quite complicated. Let's say, okay, this is like, for example, here is the middle, and then the membrane has to extend in this direction for this and this long, and then because of that, this, for example, here has to be a transmembrane helix, something like this. But we just do not directly see it. So in the end, if you want to put it this way, this is also a prediction or an approximation, at least, of the truth, even if there is only one truth. Yeah, and they have different approaches, and we do not know which of the two is better. We just cannot really tell, yes?
But just to add one point. So what they actually do is for every single residue in the 3D structure, they say this is a membrane helix or not. That's the assignment these two methods do.
Yeah. So since we don't...
How do they know?
Yeah. Sorry?
How do they know that it's membrane helix or not?
Again, I cannot tell you the algorithms. They are more complicated than you would think. I would have to read the papers again as well, but
to simplify it, really, if you went back one, so I will get back in this structure. I will get back to this issue in a little bit later, maybe next week, maybe today. So the interface between solvent and membrane is interface between two different environments. So if you assume that the membrane layers somewhere, the top is here and the lower one is most likely somewhere in this angle here. That's the way this molecule is oriented. So usually I show them like this, but this most likely is a little bit of an angle. And so first of all, without looking at anything, you sort of see somehow the ends of the helices. Okay? And helices typically end in the end of the membrane. So without knowing anything, you sort of get a guess. Okay? That gives you some idea how these methods work. And the next thing is when you look at more detail which residues you have, you see something that people describe as a hydrophobic cuff. So there is an end signal that distinguishes, in terms of this residues that I use, that distinguishes this slightly from all the others. That's what they try to pick up. in more or less elaborate ways. Actually, very elaborate ways.
Yeah, like there's many more. I know PDBTM, for example, also looks at the actual assembly. So we are in the structures, we always get something like this, but this is not how the protein is actually in the membrane. I believe in this case it builds homotrimers, so 3 of these go together, and this is the structure that then actually is in the membrane. Again, in this case, it might not change too much. It's a fairly simple transmembrane protein, but this can also have an effect. And all of this you have to take into account and try to make your best guess of where the membrane is located. Okay, right, and since we cannot really know which of the two annotations is better, we just have to assume both of them correct. If a prediction method predicts something that fits PDBTM, or if it predicts something that fits OPM, both of those are equally good to us in the end. Because we don't know, and the algorithms might be biased, and we simply cannot say one is better than the other. So if it predicts according to either of the two, it's correct. And just to wrap this up, this is what the same protein would look like if we highlighted the annotation, in this case from BDBTM. So red and blue are the inside and outside of the membrane, and then yellow is everything that is annotated as transmembrane region. Yes, question?
So you said before these things were grouping in groups of 3 or something, right?
Yes, for example.
Okay. Is it like the place where the stuff comes in and outside of the membrane is between these 3 or inside one of these?
In this case,
yes.
It's...
Yes? So,
It is either there or there, yes.
Ah, okay.
In some cases, both.
Yeah.
So what is symbol of biology? So you have examples where the multimer formation is exactly where the pore sits. You have examples where I showed you some, I may show you some of the structures later, where the pore sits in essentially inside of these cells. This is a bacteriolopsin. And you have examples where you have different activities going through the pore of the meta solution and the individual one. So that was a full yes on your question.
Okay, so now we have for every residue an annotation, this is transmembrane helix or not. And now we can evaluate this. So what we can do first is look at it on the residue basis, right? For every residue, do we predict this residue to be transmembrane helix or not? Is this correct or not? So you get your typical scores, true, false, positive, negative, right? So on this you can calculate these 2 scores, the recall and the precision. So this is all residues that are correctly predicted as transmembrane helix divided by all residues that are transmembrane helix, and this is all that are correctly predicted as transmembrane helix by all that are predicted as transmembrane helix. Simple recall precision. So let's plot this. This is 12 methods now. This legend will stay the same, so once you've memorized it, you're fine. It is sorted by the publication date of the method. So TopRed 2 is the oldest one. MEMSAT SVM is the most recent one from 2009, I believe in this case. And then in gray are our 3 baseline predictors that I mentioned at the start, right? So here we have our 2 scores from the slide before. Again, this is recall, this is precision. And first of all, without going into any details here, you can see that they kind of are all the same in a way. So they don't vary too much. And this isn't too surprising if you think about it, because on a residue basis, single errors aren't punished too much, right? Because you have lots of residues, and in the end it kind of averages out if you just get roughly the helix correct. And so that is one thing that's kind of unsatisfying with these scores. While we're at it, looking at random, we can see that they perform much worse, so that's good. If they didn't, we would be in trouble, right? But we can also see the simple hydrophobicity-based prediction is extremely good, which is astounding, or not, however you think about it, but this just shows that hydrophobicity is a very strong feature, even with the most recent data that we have today. I mean, it gets close to the worst prediction method here, right? It's really good. Right, so what else did I want to say here? Yes, the next point is, on a residue basis, you would not punish anything like this. So this clearly does not make any sense as a prediction, right? There is no helix of 1 or 2 residues. To be fair, no method would give you a prediction like this, because even if the machine learning model would give this as an output, there would be a manual filter afterwards that says, okay, this does not make any sense, let's just kick it out. But still, technically, the score would not capture this. Carlo first.
Can you go back, please?
Yes.
So I have maybe a silly question, but we notice here that hydrophobicity index is showing almost the same result for all these methods.
Yes.
But could this be even because these methods are based on hydrophobicity? Because
well, of course, all of the methods in the end are based on hydrophobicity, because that's the best feature you will ever find. The question is, how do they incorporate it? So many of them are much more than just you sum up over a window of hydrophobicity. But in the end, they all use the feature as, of course, yeah. So the closest one would be TopRed in terms of the approach. TopRed also is a sliding window. It's a bit more, it has 2 more filters and the window isn't linear, but that's really about it. So TopRed is very similar to what the Hydro predictor does here.
You may remember that I essentially, on Tuesday, the one in the room, I described the simple idea how to use hydrophobicity to predict by Gunnar van Heijnen.
Yeah.
And that is in fact the method TopRed.
Yeah.
I should have said that.
One more question or answered? No? Okay. I thought you had your hands up. Sorry. Okay. Right. And finally, there's one more important point about these per residue scores. If you say you can measure on the residue basis, this is like on the residue basis, then you are saying I know exactly here the helix ends, here the outside begins. But we just saw we cannot really do this. Even if we have the 3D structure at an extremely good resolution, we don't really know where the membrane exactly ends. There is an interfacial regions, they might be wobbling up a bit and down even. I mean, this is flexible in reality, right? So we do not know the exact cutoff, we know the annotations differ. So this is essentially based on an assumption that we cannot really satisfy. So in that regard as well, these scores are not too convincing to me, at least. So what can we do instead? We can measure on a segment basis. We cannot look not at residues, but at whole helices. And for this we have these definitions here. So now we say, okay, I have the number of correctly predicted helices and I divide by the number of observed helices. Again, this is the recall, just now on a helix basis instead of a residue basis. And I have the number of correctly predicted helices divided by the number of predicted helices, which is in turn again the precision. And now these scores you can calculate for every sequence where you have a prediction. And when you have that, you build from these two this final score, which looks more complicated than it really is. So all this says is, you have a protein, you look, are these two both 1? So do you have, for every observed helix exactly, 1 predicted helix, and do you count all of those correctly predicted? What that is, we will get to in a second. And if that is the case, then you put a 1 and you sum this up over every protein in the data set. So in the end, you get a percentage of proteins in your data set that is completely correctly predicted. This is what QOK is. And this is what we're going to look at for the next points. But first of all,
what is the difference between correctly predicted and just predicted? Are the correctly predicted manually done?
Yeah, that's exactly the point. So what is correctly predicted? I'm making an assumption here, and this is exactly what the next slide is about. So say this is your observed helix up here, yes? This is the annotation from PDBTM or from OPM, and they say the blue here is where my helix lies. Now the prediction is here, and in this case what we allow is that the endpoints deviate by up to 5 residues. So the prediction could go from here to here, for example, maximally, and we would still say, okay, the method kind of saw this is the helix, and in a way this accounts also for what we just said, that we cannot really say where exactly things end and begin. So yeah, this kind of takes care of that. But of course, we also have to say only one predicted helix can map to one observed helix and the other way around. This is almost impossible to do otherwise if you have this endpoint criterion, because you kind of, you can't move them that much. But in the past, what was often done was that if you have any overlap of 5 residues, for example, then it's correct. And the problem is then you can have helices that are aligned like this, and it's still counted as correct. And that is a very big difference, because like if you have a protein like Proteurhodopsin that we just saw where the loops are very short, then you are already in the next helix pretty much if you have only a small overlap. So this is a much more stringent criterion for a correctly predicted helix. And yeah, with that we can predict the scores we saw before. Yes?
Stay on this one. You said it. I just want to be more explicit about it.
Yes.
The helix is counted only one. What it means is, so you see the maximum length of the helix that is predicted that is correct would be from here to here. Now you could imagine that the next observed one starts here. And you could imagine in this particular case here that you in fact predict a very, very, very long helix. And it would be counted twice right, because it has the maximum overlap to this and to that. And that is not possible here. And that, so far, most people implement this algorithm forgetting that you shouldn't count twice. And that's the important point. That's an important point.
Yeah. The point why we're at it, the problem with the number is that in transmembrane proteins, the exact correct number makes a big difference, because it changes where your protein starts and ends, on which side of the membrane. And functionally, this is very important. So even if you just get one wrong, this is a very important difference. And this is why this score QOK is so strict and says you have to get all of them correct. It's all or nothing. It's still interesting to see if you have always 4 out of 5 correct, that's nice, but for a very useful result where you can actually say, okay, now I know something about the protein, I would argue you have to get all of them correct. And this is why QK is such a strict score. Okay, so let's look at these results. Again, same methods. Due to time constraints, I'm not talking about the dataset collection in detail and so on, but the full dataset has 190 proteins. And if we look at the results, we can see that the methods vary much more now than they did before. We can also see it's much worse than before. So on average, we have around half of the proteins completely correctly predicted, roughly. Around 50% is where they all are. And some of them, like for example polyphobus, stand out as particularly good, or memset SVM, while others do not so well. The error bars are approximated via bootstrapping and single standard deviation. And if we take those into account, we can see that, for example, all of these pretty much cannot be distinguished. And that, of course, is also an effect of our small data set. We simply do not have that much data, so it's very hard to say there are significant differences between different prediction methods. And again, looking at our hydro predictor, it's even better than the worst method here. So the trend holds that this is so this is so good. I was convinced I made an error somewhere, but I cannot find one. It really is that good. Somehow, hydrophobicity is just an extremely strong signal. Okay, so this is for the full set. Now we're going to split the set up into several subsets. Here it is split up by what I call new and old. So what you do is you have your 12 methods that you are evaluating, and now you collect training data. So in this case, this actually worked. It's not always available, so you have to be a bit lucky. But here I could collect all of the training sets, and then I compared this to my evaluation data set. And then you have, and then you see which sequences are similar to what I have in my dataset, or even the same, and which ones are not. And all that are not, you say, are completely new. The methods have never seen these sequences because, of course, since the last method was published, new data has been released, and it might be homologous to something they have already seen, or it might be a completely new protein that stands on its own. And this is the new part, and then the old part is all they've seen before. And as we can see, the difference is quite large. Now, the first thing that might come to mind is you say, okay, this is just overfitting. They overtrained their data immensely. And that is kind of true, but there's a bit more to the story. So if you look at the leftmost bar here in red, top pred, again, you just heard what top pred does pretty much. It's just summing up hydrophobicity values over a window. There is no real training in there, right? You have, I believe, 1 or 2 parameters, if at all, and they are manually set. This is not really machine learning. So this method, yes?
They were set when 146 is the set of things that are known for the right-hand side.
Yeah.
Burkhard used one of those.
Yeah.
It's just so old that none of them were available.
Yeah. Well, that's of course the next.
Really is not biased by any of these two.
Yeah. So of course this old is mostly, so this is the common denominator, right? And memset SVM is the most recent method. This is pretty much what defines the set almost on its own because it's the most recent method. It used most of the data. And top pred really had nothing to do with this, and we can see, but even top pred is worse, right? So it can't just be overtraining. In fact, you could argue that the difference that top pred has, which is 25 percentage points between new and old, is not any kind of overtraining. It is actually the new insights we get from new proteins, right? They are all transmembrane proteins, but they might have new signals, different conformations, things we simply had not seen before. And this is what is also causing the decrease in performance. And this is, in this case, also mirrored by the hydro predictor, which again is quite similar, and it also behaves similarly. So this kind of supports that idea. And then with that in mind, you could say, okay, anything that is more than this, more than 25 points, is this. There we can say, okay, this might actually be overtraining. And it's a bit hard to see here, but if we take the difference between them, we can see that in particular the more recent methods tend to lose a lot on the new data, so might have significantly overtrained their training sets. And finally, you can see Scampi here is now the best method, and this is also very reassuring in a way, because Scampi is that method I alluded to in the very beginning. So I'm not sure when it was published, I believe 2008 or so. So it's certainly at a time where everyone was already using neural networks, hidden Markov models, throwing all of the machine learning on it that you could possibly do. And then they published this method, which is like a glorified top pred. It's a hidden Markov model, but it's a very, very simple hidden Markov model. It just trains 2 parameters. And in the end, it's also just built on very recent knowledge of hydrophobicity values, of the energy that is required to insert a segment into the membrane. So that was an experiment that was done shortly before. There was data released from this about the required energies, and they used this in their prediction method. And as you can see, they are performing quite well. So if this really is to a good degree due to overtraining, then the fact that Scampi is not affected makes sense, because it couldn't really overtrain. Okay, this I all said. So next, the same set, we put it together again. We have 190 proteins again, and we divide it now by the origin of the sequence. So the protein we're looking at could be from eukaryotes or could be from bacteria. And this is the performance for these 2 subsets. And as we can see, eukaryotes are generally predicted better, which actually is contradicting previous results. So typically eukaryotes were always the ones that performed worse. And we don't really know why this is the case. And to be fair, I don't have much more to say here. I just want to show you this. So there is a difference, clearly, but I cannot really give you an explanation as to why that is. And I believe, one more final point. So in this case, it does not seem to be an effect of how new data you had available, because PhD HTM, which is the orange one, and memset SVM, which is this one, behave almost similarly between the 2. So apparently this has nothing to do with overtraining really. It's just, we don't really know. Maybe it's just a bias in the data set even. I can't really not dispel that, but there's also no other explanation. At least I don't have one. Was that a question? No. Okay.
I have a question.
Yes. Sorry.
From this slide here. What do you mean overtraining? Aren't those trained by their own data sets separately? So if there is an overtraining, there should be on both sides, right?
Yes, so you could say one method trained only on eukaryotes, for example, right? And then you would expect it to be a bit better here, but.
So you would actually use some parameters from those eukaryotes onto the bacteria.
Yeah, of course. So I don't believe there is a method that says I do only predict eukaryotes. So they just say give me a sequence and I predict, and they claim.
There is no method.
Yeah.
But let's get back to this. I believe there is some puzzle here, something else to be done and to be said that we don't actually know. But the fact is, historically, all the membrane proteins that were known were bacterial. So if there is any bias with respect to training, it's on that side. The newer structures are on that side. Fairly new that we can actually do eukaryotic structures. New is defined as less than 10 years.
Okay, that we said. So the final split of the data set is now by the number of helices that are actually observed in the proteins that we are looking at. So we have many proteins that have just one helix, for example. Also in reality, this is the biggest class of proteins by far, membrane proteins. And we can see that on those the performance is quite good for every method. And as soon as more helices appear in the protein, performance decreases. Now there's two possible reasons for this. One thing is you might say, well, QOK mandates that every helix is correctly predicted. So if you have more helices in the protein, then you would expect it to get harder, right? Because you have more chances to mess up. You have more things to get right. So clearly performance should probably decrease if the problem in a way gets harder. But at the same time, this could also be an effect of the biology behind it. So maybe proteins that have more helices really are more difficult to predict. For example, as a very basic point is that these proteins here, which only have one helix, the hydrophobicity signal in that helix is typically much stronger. While when proteins have many helices, the hydrophobicity signal in those helices tends to get weaker. One of the reasons is probably because they cluster together in the way they shield themselves, so they don't need it that much, if you will. But this, of course, could trip over the methods, right? If they are looking for a very strong hydrophobicity signal, then the signal here is weaker than it is over here. And in the end, we cannot really divide between these two effects. So we don't know if this is, probably it's both. It's both statistically more likely to get wrong, but it's also different data. And saying how much comes from which part is, very hard to do. Yeah.
Like a few slides back, you. It's okay.
Yeah.
No, you can show this.
Yeah.
You said like it doesn't make sense for a protein to have one helix.
Did I? I didn't mean to say that. Protein. So again, most transmembrane proteins in humans.
Oh, that's the slide of the CHHC.
Yeah, exactly.
Oh, yeah. So okay. So what's the question on that?
The confusion between residue and segment, this one.
Right, so this is one residue helix. So, how do I say this? So, let's look at the protein. Right, this is a transmembrane helix, yeah? Here I'm not displaying single residues. This is the typical cartoon representation because it looks nice, you see the secondary structure. But this is composed of roughly 20 residues. So there's an amino acid here, here. It goes all the way up the helix, yeah? And what I'm saying is doesn't make sense if you predict a helix that is just one residue. It would be like one turn of this. That doesn't make sense. But proteins that have one single helix, that is actually the most common class in human.
One turn of this would be 4 residues.
One bend, I meant. Yes, you're right. Okay. Does that make it clear, or?
So in this you mean one helix of maybe 20 or 40
Yes, exactly.
residues.
Yeah. So yeah, right. So TMH is transmembrane helix, not residue, but the whole helix. Okay. Yes.
Some proteins have, in fact, most proteins that are associated to the membrane that we know have one membrane helix.
Yeah.
There is a variety of reasons for that. The proteins that we care more for, in some sense, are the ones that have more. Everything that sits on the lower part of this graph here is what we care more for.
Yeah. So to give an
Actually, here the numbers are different. Why do I say that most proteins that we know have a single helix? It's not reflected by your numbers, right?
Why? It's 92 proteins. So yeah, well, it doesn't. It's not exactly like it would be in human. And the number of actual helices.
Oh.
And it's the okay. So maybe I should have. This is confusing. So okay, we have 92 proteins in here, yes.
Okay.
And they all have a single helix. And this is double the number because of OPM and PDBTM. So maybe I should have just taken those numbers out. Sorry if that was confusing.
But again, so here in this particular case, it's also falsified. But this is a sequence unique subset. So if you add up 92 and 60 plus 33, 60 plus 33 is 93 versus 92, and you immediately verify that what I said, that most have one, is wrong. In this particular case, most actually have more than one. But the reason here is the way he filters out the data. If you just look at the PDB and pick out every protein that has a membrane insertion, and then we ask how many membrane helices to have, then most of those, unfiltered, have one. Here is a sequence unique filtered subset that picks out the differential ones. Here the story is slightly different.
Yeah. So this in no way does reflect the real distribution, and as we just heard, so for example there's a class that has 7 transmembrane helices, GPCRs, and they make up by far the majority of drug targets in humans. So those are extremely interesting to us, and they are also quite common in human, but yeah, so it's not a linear distribution is what I'm saying. I don't have a picture here, unfortunately. So 1 is always the highest, and then depending on the organism you have a few spikes. So 7 in human, in bacteria I believe 4 is also, and 12 maybe, but yeah, sorry.
You didn't touch upon the fact, so there is an immense fall here that you see, right? And some of that fall is absolutely trivial. Okay? How can you see that? Well, you could, for instance, so you could call this simple hydrophobicity sort of random prediction, or you could look at random predictions, and they fall too. But this is the first, and essentially the point is you have a protein in which you know there's going to be a membrane helix. Find it. That's your task. Okay, so simple hydrophobicity finds it most of the time somehow. It's a simple task. While here you may have 5 different ones. And now the task is much more complicated, okay? And that is, so here we have a data set of proteins that all are known to have 1. Just find the one with the highest hydrophobicity and you'll get it right. Here, it will be between 2 and 5, and where they are is more complicated. And that's why it drops, and the more helices you have, the more complicated it is to make them at random. And that's exactly what you see. Now the trouble is that it's not easy to come up with a score that sort of completely subtracts. You could look at the difference to random, and Jonas looked at all of these scores, and none of them really was completely reflecting the truth. So there is no simple way to show that in a more intelligent way. Or we did not find a simple way.
Yes, and they were contradicting themselves, so not very convincing, all of them. Right. So all this time, I touched upon this before, all this time we've looked at QOK. So every helix had to be correct, we needed the correct number of helices, and they had to be correctly placed by the schematic we looked at in the very beginning. Now, just to give you some idea of what the background between this is, here we just look how many helices are predicted and how many are observed. So ideally, we want the same number here. We want every method to be at 0. That still does not mean that they are at the right position, but having the right number is a good start, at least. And as you can see, actually most of them are at around are at 0. So a lot of the time the methods get the number of helices correct. And if you take just minus and plus 1 into account, these parts here, then actually for every method this is more than 90% of the data. For most of them even more than 95% of the data. So in a way, quite often it's just one helix that's breaking it in a way, or the location is wrong. But you might say it's not so bad. Then again, as I said, one helix can make a very big difference. So I wanted to show you this, but at the same time this should not be diminishing what we saw before, what seemed like much worse performance. If you miss one helix, you miss one helix, and for transmembrane proteins that is very severe. So yeah, that's all I have to say here. Finally, all this time we had a set of proteins. We knew they were transmembrane proteins. We gave them to our methods. We told them, tell me where the helices are. But before you do that, there's another problem. You actually have to see, is something a transmembrane protein or not? So now we have a set of soluble proteins that are not transmembrane, and they are from eukaryotes, gram-negative and gram-positive bacteria, and we give them as input to the methods and see what they predict. So some of these methods actually have a separate filter that you execute first, where they decide, okay, this is transmembrane or not, and then you do the prediction. Some of them, like TopRed for example, don't have this. If TopRed predicts a helix in these proteins, then it thinks it's a transmembrane protein. And as you can see, due to this, TopRed, for example, gets most of these proteins wrong. So you cannot use TopRed to distinguish if something is a soluble protein or a transmembrane protein. For 2/3 of the soluble proteins, it would say, yeah, this is transmembrane protein. I found a helix here, because that's simply not what it was built for. And then you can also see there are some methods that perform quite well. So this is false positive rate. We want this to be as low as possible. TMHMM2, for example, which was published around 2000, I believe, certainly is quite old, is doing fairly well. So this method kind of can see the difference. But now we get into the next important point. This data set we are looking at now are soluble proteins, but they do not have signal peptides. And I'm not sure how much you've heard about this before, so just very quickly, not at all? Okay, so the signal peptide is like the signal to the cell that something should be transported somewhere. And there are different kinds, and the problem with them is that they have 3 regions roughly, and there's one region where it's extremely hydrophobic. And if your method does not know that this exists, then it sees, oh yeah, look, the hydrophobic region, this is a transmembrane helix. So this is like it was made for getting misclassifications of transmembrane helices. In fact, to make it even worse, sometimes the signal peptide is a transmembrane helix. So often they are cleaved off, but sometimes it's even left on and does become a transmembrane helix. So this is a distinction that has to be made by these methods. If they don't know about this, then you have a big problem when distinguishing between soluble proteins and transmembrane proteins. This set did not have signal peptides. Now the set has signal peptides. And now all the bold methods here, so these and Scampi, are the ones that do not in any way account for the presence of signal peptides in reality. And as you can see, all of them perform horrible. Which is exactly the point. So if you do not know about this, you will make misclassifications. And just to put one example, TMHMM2, we saw before had like 1% false positive rate only on the set, and now it went up to 20 and more. And Phobius is in a way building up on TMHMM2. It's the same model, but it accounts for signal peptides. And look at what different this makes, right? So this gets it almost back to the performance we had before. So yeah, as I said, you have to account for signal peptides, yes.
Can you explain signal peptides again?
I'm sorry?
Signal peptides again.
What about signal?
What are signal peptides? How do you explain?
Where they are in the protein?
Like, difference between signal peptides and no signal peptides.
I'm not sure.
Just quickly get that. The signal peptides. So there's I showed you eukaryotic cells, and there's subcompartments. There's a nucleus, and there are other subcompartments. A bacterial cell essentially has an outside and an inside, and the cell membrane. That's what it has. Now, even for a bacterial cell, some proteins go to the outside. I showed you some numbers that many proteins, in fact, are secreted on Tuesday. Secreted means put to the outside. So some proteins go to the membrane, sit in the membrane, and some proteins go further. The recognition how to bring proteins out there is a transport mechanism. And one way in which this is by recognizing a part of that protein. So there's a protein that comes and recognizes that the begin of the protein, the N-terminal end, in fact, has some signal. That's why it's called a signal peptide. It recognizes that signal. This signal can extend to something like 50 residues, it can be 20 residues. Shuttles the protein through the membrane or into the membrane and then further through the membrane. And that's what Jonas just said. In some cases, this very same thing that typically is very hydrophobic. So to prediction methods, it looks like a membrane helix, is inserted as a membrane helix in the membrane. Sometimes it's just cleaved off. Cleaved off means there comes another protein that really is like a scissor, so-called kinase, that cuts it off, and then it's just getting through the membrane. Those are the signal peptides he's talking about. Is that answering your question?
Okay, sorry. Right, and then one more thing, you can also see that the scores are typically worse for gram-positive, for the sequences from gram-positive bacteria. And one explanation for this could be that the signal peptides in gram-positive bacteria are typically longer and more hydrophobic, so it makes the chance to misclassify even worse. However, this isn't really the whole story. So if you look at why these are misclassified, you can see that for the other data sets, yes, quite often the signal peptide and the predicted transmembrane helix overlap. So this is what caused the problem. However, for the gram-positive set, this is often not the case. Often the helix is predicted very far away from the signal peptide. So there must be more to this than just misclassifying the signal peptides, in particular for the gram-positive sequences. Okay, and finally, looking at the other side of the coin, you have transmembrane proteins. Do you even see this as a transmembrane protein? Now, top red 2, 100%, of course, you have to see this in conjunction with what we saw before. Yeah, I mean, top red predicts almost everything as a transmembrane protein, so yes, of course, it gets 100%. But this doesn't mean this is the method you should be using. So, generally, all methods are pretty good at this. Again, hydrophobicity is a very strong signal. It's not too hard to recognize. If you combine all of these tables, which I can't really put on one slide, but I'm going to tell you, if you combine them all, something like Phobius would be your best choice probably. It gets almost all of the transmembrane proteins correct, and it has very low, around below 5% false positive rate on the soluble proteins, and this is one of the methods that does account for signal peptides. Right, this is a summary of everything I said. And then just one final thing to give you an idea of the future. So a friend of mine has been working on a new transmembrane prediction method called TM-seq in blue here, and this is comparing it against Polyphobius, which was pretty much the best method from the previous evaluation, and MEMS@SBM, which was also very good. And as you can see, with some intelligence and machine learning and even more recent data, we can even improve upon that and also transfer this to the topology. So this is something I didn't touch upon at all, but say you have the helices at the correct location, then the next step that you typically also take is you predict, are my non-helix residues, are the loops on the outside or the inside of the membrane? And this is what's typically referred to as topology. And the Q top is just this. So the QOK is 1, the protein is correctly predicted, and then you also correctly predict the inside-outside topology. And the performance of this method transfers quite nicely between the two, so also having to get topology does not really diminish the score. Okay, but this just as a short notice, it's not published yet, but it hopefully will be soon. This is not supposed to be here. Thank you. Any questions? And a comic.
I have one question.
Yeah.
About,
Back or?
can you go back?
Yeah.
Somewhere again? Sorry.
Yeah.
Where there was the single prediction and the more predictions and.
This one.
Yeah.
Yeah.
Is there any way you can say you can get the more predictions by combining single predictions? Like,
I'm not sure what you mean.
If we're good on one, is there any way we can recognize these different single ones in a chain of 3 or so?
Meta methods.
Is that what you mean?
Take the best of both worlds.
So you want to take a method that's good at recognizing one helix and methods that are good at recognizing more? Or, I mean,
use the fact that we're good in recognizing one to recognize 3.
well, so the methods predict however many helices they think they see. And you...
Very simple answer. You should be able to, but so far nobody has succeeded. I don't quite understand why not. But the reality is we failed. It's the same reality if we went back to other slides. So Jonas presented a couple of examples. So some methods are better at eukaryotes, or sometimes for eukaryotes you do better, sometimes for more single helices you do better, sometimes for single peptides you do very poorly. And somehow there should be a way to find a path through these landscapes, not no way, but so far nobody has found that. There were methods that sort of tried to combine methods, but they don't find the best path through this. Why not? That's what I meant. It remains a puzzle. Should be possible, but path has not been shown. So if there's no further question, then thanks.
Thank you.
These talks are your talks on the internet already, or no?
No, I'll put it up now.
I will merge it ultimately with my slides and then replace. So Carlo will replace today's slides by the ones that merge in Jonas' slides. And sorry that we didn't do that before. We just didn't get ready in time. So I believe, Jonas, you can, yeah, thanks. Okay, so just a quick recap. Jonas showed you, so that's the lipid bilayer. Membrane helices are inserted into this construct made out of two groups of lipids. Head groups are soluble in water. This is lipid, this is hydrophobic, this is not, this is oily. Is 2.5 to 3.5 nanometers long. So the span of the membrane that sits around the cell, and it creates a different environment. Typically inside of a protein you have hydrophobic residues. Now this inverts this inside outside. Outside you have water soluble residues. That's how a typical protein looks. A membrane protein must be somehow just the opposite, because these helices sit in an oily environment. And there are different ones. And again, I said on Tuesday that you can recognize the cuff sometimes just looking at these images. But as Jonas again pointed out, you cannot see the lipid in the crystal. So you can recognize it. You see in this particular case, you have this sort of mushroomy structure, and sometimes you really see as if you squeeze something, and outside of the squeezed part, you see the outside world in some sense. And I said that most comparative modeling for membrane proteins are very poor. It's this tiny blue here, and that is because the experimental structures are so difficult to do. Today we have about 1,000 structures, but about only 100 families. So 100 structures are somehow unique. That's all we have. Out of the set of 100,000 structures for globular proteins, this is what we get for membrane proteins, despite 60 or 50 to 60% of the drug targets being membrane proteins. And the methods for 1D, I showed you various ones, and he talked today, are very successful. But Jonas also showed you that there remains something to be done. The frustrating part is many groups have tried and have failed. The pressure is that I also showed you examples how you do cryo-EM, cryo-electron microscopy. In cryo-EM, what you try to do is you essentially look with a microscope at a protein, and I showed you what you see is just the surface. Now what people do is they do modeling inside of that surface. So they say there are components in there for which I know the structure, I can put them in. And that is particularly interesting for membrane proteins. So you could imagine now with a good prediction of where the helix sits and how the orientation is, and some sort of surface image, you may get to the point of knowing how the protein looks. But it turns out that for this to work, you in fact need a better prediction of the helix, the membrane helix. So today's methods are still not good enough to sort of make EM plus modeling sufficient. So that's why the incentive. Although everybody who has tried somehow produce something that is similar, as Jonas showed you, over 2 decades, the pressure still is on to get some money. Today I'm going to talk, not today, for the remainder of the time, there's not much time left. I could actually do that on Tuesday, or no. Let me just go through. If you stopped here and asked questions, we could call it a day and do 15 minutes of question and answer. Else, I'm going to begin this topic and continue this topic here next Tuesday. The call is yours. Okay, so there's a different type of membrane proteins, the beta barrel membrane proteins, and most of the slides come from Henry Bigelow. Again, and here's a bunch of these examples. In all of these, you do not see in this crystal structure the membrane itself. Those 2 lines here indicate the lipid layer. You do see, you may by now recognize that what you see here are beta strands. So rather than now having the helix rod go through the membrane like this, you essentially have the fingers of a hand beta strands at a 45-degree angle. And those are in fact here, in this particular case, 11 different structures. And all of them you see essentially the traces that you see, the lines that you seem to make out here are sort of 45 angle-ish. They're roughly looking like that. Okay? And now we have an example. That's another point I should have brought up on the slides before. Let me just go back on these slides here. The question that you had. So the function of a protein typically has to do with a particular aggregation of the same thing again and again. Here is an example for that. It's a dimer. Dimer means you have twice the same proteins the same protein that comes together. It's completely symmetric here in this axis. Most proteins act in the cell as aggregates of multimers. Mostly, the way they aggregate the multimer is exactly responsible for the function. So if you do a dimer 2 or tetramer 4, you do not get the same function. Some proteins, in fact, do function differently in different aggregates. And this is back to your whole story. In some holes, you absolutely need a particular way of aggregating them. Now this I happen to not be able to show with that slide. But today we perceive that the way it aggregates is responsible for function. It seems over the last sort of 5 years, we pull out more and more data that the same protein can aggregate in different ways. So historically, again, determining a 3-dimensional structure, that's the only way you can really see that, was not easy. So you were happy when you got one. So now the experimentalists are a little bit better at that today. And now they see that, in fact, sometimes they get crystal structures from different ways of aggregation. So they have dimers, they have trimers, they have tetramers of the same protein. Sometimes, in fact, it turns out those are all biological, meaning they exist somewhere in a cell. And sometimes some of them are only happening in the crystal, for all we know. How many of these will be possible if in 10 or 20 years? We don't know at this point. At this point, what the fact is that we believe much more now that they can aggregate in different ways than we believed 10 years ago. So 10 years ago, it was much more this idea, there's one way. Today, there seems to be different ways that you can influence by slightly different environments. Okay? Still, they're relevant for function. And that is in particular here for the porine type of barrels. Again, what we see typically here are already aggregates. These porins sit in the outer membrane and they typically form holes. Unlike the helical proteins that we had so far, in the helical proteins typically transport is an active thing. So you have a hole. If you have a hole, typically it's smaller, and if you have it, it sort of opens and closes according to something that happens in the environment. So the right ion comes, or some other trigger triggers the import. These are essentially like holes that are open. Relatively big and relatively open. So how come that the cell is not wounded by having an open door?
There is lipid blocking in the metabolism.
Well, no, it's really, it's the lipid, so they sit, so if in these images here, that would be the, well again, so now I have a problem to show, my fingers just cannot do that. Okay, this is the lipid, and now I take it out of the lipid here and I show you like this. It's like this, so you can see through it. Yes, there's lipid all around it, but in the middle here, and again, so it's a 45-degree angle. I'm changing the angle so that you can better look at it, because my fingers can't do that. But so it's open.
You need peptides.
You can put peptides through. Yes, you don't. Actually, at least those that I show here. So you could imagine that you have a lid. Actually, that does exist. But many of them don't even have a lid. It really is a hole. How would you do that? Again, I kept telling you, imagine this like a medieval village, and you put a wall around it, and you put a guard in the gate, and the guard, now we have a medieval village where you have an open gate. What would you do?
Extra guards.
Extra guards, but that's a great idea. But that's in some sense his idea, right? And again, there are examples for that. But now assume that I'm lacking the right image to translate it to my medieval village now. But let's assume, because of some reason that cannot be. Here's the story. And let's assume that time doesn't matter. We have infinite time, but we cannot close the hole. So a cannonball shot a hole into the wall, and that was a golden cannonball, and we don't want to destroy the hole. Whatever the reason for that. This is a stupid story. And I have ample time. What would you do to protect my village again? So there's one hole in the wall.
Fix the wall.
Yeah, that's what I'm saying, is you don't fix the wall. So that is not part of, you have ample time. My story is really, I have to come up with a better image. But imagine a situation in which you cannot fix the hole.
So whatever comes in should go out again. Maybe that's...
Yeah, so but that is what he says, right? You have to have somebody to kick them out again. Like,
Sure, there was always something flowing out of the hole, so there would always be resistance to something coming in. Like, I don't know, you look up a giant hose.
No, in this particular
water is always coming out of the hole.
Yeah, that's what, essentially you have the same idea here. Okay, I have to give you the solution that I have in my head. It's too far-fetched. Actually, you build a second wall. As simple as that. My story, of course, doesn't fit, because if you can build a whole wall, you can clearly fix one hole. But in this particular case, this is exactly the way it's done. And these, in fact, sit in bacteria that have two membranes. In the outer part of bacteria that have two membranes. They also sit in human, but only in mitochondria. In mitochondria, again, I told you, is a relic of evolutionary times where we imported bacteria into our body. And today, in every single cell, there are hundreds of mitochondria, but they're all sort of from a bacterial past. Yes?
I didn't get this. Is there already a second membrane or it's just built around?
Yes, there is. Well, so
Now, but my image is misleading, but the answer to your question is also, or your question in some sense is also misleading. In an evolutionary sense, there are bacteria that have two membranes. So most likely the first bacterium had one. And then some species evolved this idea of having a second membrane. So now I would call that build a second wall around. And then in the second wall around, and this is why the story sort of gets misleading, is into the second wall you can now afford to have more open holes. So you still don't have a full transport of everything, but certain things that fit this, at least into the second wall, they can get in equally, simple, without any further gating. And then you hope that the further gating is done inside, but that way you already get closer to the cell, some stuff that you care for. In a simpler way that, again, active transport translates to using energy. Active transport means that every single time you get some sugar into the cell, or nutrient, or some material that you need for the cell to live, you need to invest energy to just get it in. And this is in fact what most membranes do. Import requires energy. And that's the beauty here. It's completely passive. Doesn't take anything. This is the way they look, and here are some of the functions. There's a variety. They're mostly referred to as porins because they essentially are a form pore in the membrane. And again, here's the membrane. This is the way they look. Now, Henry's most important part of the work came, in fact, by taking the structures, 11 unique ones he had it when he started, very small number, and label different states. How do you do that? Well, you look at the three-dimensional structure here, and you in fact, no, I don't have a per residue trace of a membrane, and you say this is where the lipid ends, this is where the lipid ends, and this is the strand that goes up, and this is the strand that goes down, because they always go like this. So you invent a method that essentially labels 3D coordinates according to the membrane that you cannot see. And that is in fact, he developed that method at the same time as these other people developed their database, and ultimately his signal was the hydrophobic Huff signal. And that simply, ultimately he did it all on the screen. He had just a limited number, but what he did, because he was a mathematician, so he began with an algorithm, and then verified on the screen that the algorithm did something that is reasonable. And again, as Jonas already said, there's some variety, but you know, that is the beginning of the label. Once you have this labeling, so up and down strand on two different sides, you in fact think or can consider that this is the type of problem that you can solve with a hidden Markov model. You have a grammar. You have states, you have two different types of strands. They alternate in the way they look, every other one. You have a certain periplasmic on the inside of the membrane, and on the outside of the membrane you have this loop that goes out and back in, loop that goes out and back in. They look slightly different on this side than on that side, and you could possibly model all of that, right? How do you find out whether, if that is the idea, and those are the labels you get by just looking at structures, how do you verify that this model would work? How do you fill these states, if you believe there are states? Labeling means you look at a structure and you say this is an up strand because I can't see it, right? See it on a computer or see it on the screen, it's the same thing. And then you have a down strand and so forth and so on, and that is your model. It's an HMM model. So far so good. How do you find out whether it makes sense? So the first step, of course, is that you have to put the data into the model. So now that you have this label, you will simply look at every single residue that belongs to one label, and we'll plot the probability of certain amino acids. So the height of the letter here is essentially proportional to how often you see that at that particular position. So this is the strand going through from residue from the first to the last. That's an up strand and a down strand here, and is this side and is that side of the strand. going through like this. One thing you absolutely see is there's a difference, right? You clearly see that the distribution of letters, visually, it takes you no time to see that the distribution of letters is not identical. Now the next thing you have to convince yourself of, and that I'm not going to show you on a screen here, is that this is not a random thing, but if you slightly change your data set differently, or slice your data set differently, you would still see a difference, and that this would not suddenly look like that if you took only half the data or 1/3 of the data. If you convince yourself that this is true, essentially you're ready to believe that the hidden Markov model that uses these states makes sense because you have distinction, and the next thing to do then would simply be to see how well it works. And with that, I leave you for the weekend. Thank you for the attention.